Okay, so this is going to be a long narrated PowerPoint. I may stop it at some point, but I'm not sure that there's a good place to kind of break it into two pieces. So bear with me. So what we're talking about now is going from gene to protein to how do we know which genes to use? You see, cells, be they prokaryote or eukaryote, need to regulate the gene expression because different genes are going to be needed for different conditions. In multicellular organisms, different cells are going to need different genes. And so there's a lot going on with how cells regulate their genome, how they know which books to pull off the shelves. And RNA is going to have an integral role in this. From gene to protein, in the last chapter, we talked about all the different kinds of RNAs. That is, mRNA, the transcript, tRNA, ribosomal RNA, both large and small, SNRPs, doing some of that splicing going on in the nucleus, signal recognition particles being involved in the targeting of particular proteins. Well, we're going to add a whole bunch of other RNAs involved in regulation. So we're going to have basically a list of different kinds of RNAs as long as your arm. Now, we're going to start by looking at prokaryotes, and that's because prokaryotes are relatively straightforward, relatively simple. There are some significant differences between how prokaryotes do things and eukaryotes do things. But we're going to start with prokaryotes because they're simple, and then kind of talk about how prokaryotes and eukaryotes are different and what that then means. Okay? So when we take a look at bacteria and how they're working, they can regulate what they're doing by either regulating transcription or regulating the metabolic pathway. And we're going to talk about that regulating transcription because we've already talked about sort of kind of how regulating that pathway works. Well, we're also going to talk about how the prokaryote is going to regulate transcription, how it actually gets those genes. So basically, do you need to bother to make these genes, or oh, we need these genes, we need these proteins, but we're actually going to regulate the proteins. Sometimes it's like, no, we don't even need those proteins. Let's turn that off. So, how do prokaryotes do it? Well, the fundamental thing in prokaryotes is this concept of an operon. Okay, this is a cluster, and the cluster is important here. This is a cluster of genes, all related to a single metabolic pathway that are regulated by a single on-off switch. That on-off switch is called an operon, or it's an operator, and the whole cluster of genes is called an operon. Okay, so what we're going to do is we're going to start by looking at a repressible operon. That is an operon that can be switched off by a regulatory protein. In this case, it's going to be called a repressor. Okay? Basically, what happens is that repressor binds to the DNA and blocks transcription. It basically stops the transcription. Okay? The repressor is going to be the product of a regulatory gene. That is a set of DNA somewhere else that builds a protein that's going to interact with that DNA. So what we have is a gene or a series of genes, and we have a regulatory protein that interacts with the promoter or right behind the promoter. We'll take a look at that in a sec, of the DNA. Now, the repressor, that is, that regulatory protein, has two shapes, a bind to the DNA shape and a free from the DNA shape. And in this case, that other material that binds to the protein to change its shape is referred to as a co-repressor. Now, the repressible operon that we will talk about is going to be the trip operon. This is a set of genes that builds tryptophan. Okay? Now, the gene is going to be on by default. That is, that we are going to need to make these enzymes to build tryptophan because tryptophan is going to be necessary for the cell's function. But tryptophan is also going to be a co-repressor for that regulatory protein. Basically, if there's lots of tryptophan present, we don't need to make it. So in that case, the presence of the tryptophan is going to bind to the regulatory protein, and the two will then bind to the DNA. So the repressor only functions when it is bound to its co-repressor, in this case, tryptophan. Okay, so what does that look like? Okay, tryptophan operon is on, tryptophan is present, the repressor binds to the protein, blocks tryptophan transcription. As we use up the tryptophan, it pulls off because there's no tryptophan bound to it, and we start building the enzymes that make the tryptophan. Okay, so what we have is a series of genes all grouped together. That's the sort of the fundamental component of the operon. A whole bunch of related proteins bound together and regulated by a single operator. Okay, you'll notice that we have the promoter. Just downstream from the promoter, we have the operator. So we make all of the different proteins, and they build tryptophan. Tryptophan binds to the regulatory protein, and that changes the shape of the repressor, so it now fits to the operator, and that blocks the RNA polymerase from binding to the promoter and transcribing all of those genes. So, in the presence of tryptophan, the genes are blocked, so we don't build the enzymes, so we don't build more tryptophan. But as we use up tryptophan, or we don't find it in our environment, we then pull that repressor off because it no longer fits, and we manufacture the enzymes that are going to build us tryptophan. Okay? So this is what's known as a repressible operon. And several things here are going to be unique to prokaryotes. One, the presence of an operator between the promoter and the gene. Two, that all of the genes of a single regulatory pathway are all grouped together in the operon. Okay? And then in this case, it's a repressible operon. So in the presence of the product, we are going to bind and stop the production of those proteins. Now, a repressible operon is one that is usually on and is shot off in the presence of the material. An inducible operon is one that is usually off, and what happens is the material binds to the regulatory protein and pulls it off. And so it's only going to be active in the presence of the material, as opposed to something like the tryptophan operon, which is only going to be active in the absence of the material. So that's why we have these two sort of systems. One's a repressible operon, one's an inducible operon, because the presence of the material induces the protein off of the DNA. The example we have is a lac operon, and this is an operon that produces the enzymes that are going to digest lactose. There is no reason to make these enzymes unless lactose is present in our environment. So the presence of lactose is going to induce the regulatory protein off of the operator. Okay, what does this look like? This is our lac operon. Okay, we're going to make our enzymes from the lac operon, and if there is no lactose present, then our regulatory protein is blocking the promoter and we're not building those enzymes. But if there is lactose present, then there's a reason to build those enzymes, and so the lac operon, the regulatory protein, comes off of the DNA. Okay? So that's the difference between repressible and inducible. Repressible operons are going to be off in the presence of the material. Inducible operons are going to be off in the absence of the material. In general, inducible enzymes are going to be catabolic pathways, because catabolic pathways are going to be digestive pathways, they're going to be breakdown pathways. There is no reason to build the enzymes to break something down if it's not present. Repressible enzymes are usually going to be anabolic or building pathways. That we're going to build something because we need it. We only need it if it's absent. Both of these kinds of regulation are negative control. That is, that they function to turn the enzymes, the production of the enzymes off under a set of conditions. Sometimes, however, we might want to turn something on in that condition. So sometimes we're going to have positive gene control, not an off switch, but an on switch. Okay? One good example is the positive control through a stimulatory protein. Specifically, the one that's mentioned in the book is the CRP, or cyclic AMP receptor protein. Okay, this is going to be an on switch. Okay? So why would we want an on switch? Well, sometimes we have a material like lactose present, but we don't want to spend the time to build the enzymes, that extra step to digest lactose. What happens if we have, say, a prokaryote in an environment where there's plenty of glucose and some lactose? Why would you spend the time and effort to digest the lactose if there's plenty of glucose present? So, what happens here is that in the presence of glucose, the cell's getting lots of energy, and therefore we're not going to have cyclic AMP present in the cytoplasm, because they're taking their ADP and they're easily turning it back into ATP. Because there's glucose to burn, they can use the ATP synthase process to build ATP. Everything's happy. But in the case of the absence of glucose, what's going to happen is we're going to start to take our ADP, and instead of recycling it back and rebuilding it back into ATP, we still need energy. And so we're going to start to cannibalize some of our ADP. And when we do that, when we start pulling that second phosphorus off of the ADP, we're going to get cyclic AMP. And so the presence of cyclic AMP is going to bind to the CRP, that cyclic AMP receptor protein, and that in turn is going to bind to what's known as the CRP binding site, which is just upstream of the promoter. And so what happens here is in the absence of lactose, we are going to have the regulatory protein bound to the operator. Okay, so even if we get an on signal, we've got a stop in front. Okay, but in the absence of glucose, we're going to have the CRP binding to the CRP binding site. And in the presence of lactose, there's going to be no regulatory protein blocking. And so we're going to start to make those proteins. Okay? And everything's going to be good because we're now going to be making energy from the lactose, and we now have the enzymes. All of the different operons, maltose, lactose, the other operons that are going to be breaking down different kinds of sugars are all going to have that CRP binding site upstream. And depending upon what you give that starving E. coli, lactose or maltose or some other sugar, as long as they've got those enzymes, they're going to have an available unbound operator, and we're now going to have an on switch upstream. If you think about this as sort of a train, you've got the guy in the cabin, the engineer, and he's going to go if he's got a green arrow. But he doesn't go until the conductor tells him to go. And so the green arrow is like the unbound regulatory protein on that operator, and the conductor is like the promoter saying, yep, like the CRP saying, yep, now's the time to go. If you've got a green arrow, time to go. So that's sort of the fundamental component of the prokaryote regulation. Now, let's contrast that with eukaryotes. Okay? Eukaryotes are going to regulate a lot more genes. Okay? Prokaryotes are simple, eukaryotes are complicated. One of the advantages of having an operon is that you don't need nearly as much regulation. You don't need as many regulatory proteins, because instead of each book being individually labeled, you've got books in boxes, and the boxes are labeled. And so the operon identifies a box of books. Okay? But in the eukaryote, we might want a book from this place and a book from that place because of these special conditions in this cell, or they're responding to a much more complicated environment. Okay, so eukaryotes are going to have much more precise control, and it's going to be a whole lot more complicated. Okay, so how does that work? Okay, well, in multicellular organisms, it's going to be complicated. In even single-cell eukaryotes, it's complicated. They've got lots more genes to deal with. Okay? Especially in multicellular organisms, there are a lot more individual cells, and they all have the same genes. But different cells, like your nerve genes, are going to be different expression than, say, your liver genes or your muscle genes. In fact, when cells express incorrectly, we start to have problems. So, eukaryotes are going to have differential gene expression, lots and lots of different kinds of genes, and controlled in much more precise ways. For instance, we might have the same gene expressed, the same protein expressed in a nerve cell versus a liver cell, but we may want lots more of that protein in a liver cell versus the nerve cell. And so we're going to start seeing a whole bunch more control levels. And this is one of the reasons why we get cancer cells doing things sort of incorrectly and creating problems, because the gene regulation is somehow buggered. So gene regulation in eukaryotes, very, very much more complicated, and also regulated at a bunch of different levels. We are going to have gene regulation at the chromatin level, at the level of the DNA, at the level of the splicing, at the level of transport out of the cytoplasm, where the mRNA is actually going out into the cell, at the level of translation and how long those mRNAs stick around, at the level of protein folding and processing, at the level of how long those proteins stick around, and at the level of transport to the destination, either within the cell or outside of the cell. So prokaryotes are simple, eukaryotes are complicated. So let's start at the top. Let's start at first thing that we're going to see is we're going to see histone acetylation. Okay? So what we're going to do is we're going to modify the histone proteins so that they either are available to be, the DNA is available to be read off, or it's not. Okay? We do that by adding methyl groups to the histones. By methylating the histones, we make the tails of those histone proteins sticky. So um, uh, 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 by adding methyl groups, we make them unsticky. So when there's no methyl groups attached to the DNA, acetylation, I'm sorry, it's DNA methylation, histone acetylation. By adding acetyl groups to the histone tails, we make the histones less sticky. And so they unwind, and we go from compact DNA, unacetylated, to looser DNA, acetylated. So by adding acetyl groups, we promote the unpacking of the DNA, making it available to be read off. Okay? Now, in turn, we can methylate the DNA. That is, we can add methyl groups to the DNA. If we add methyl groups to the DNA, we tend to inactivate. We tend to make those DNA less accessible to be read off. Going back to my library analogy, we add acetyl groups to unpack the books onto the shelves. But on those unpacked books, we can either put them on the shelf with the label out so you can read the book, or we can turn them around so that the label is turned around away from you. Those books are going to be much harder to find. And so by methylating DNA, we make some of that DNA inaccessible. Remember when we talked about epigenetic, some of those things where, whether or not you got the gene from your mother or father affects how it is expressed? This is that epigenetic inheritance. The methylation of DNA is fundamentally associated with this epigenetic pattern. Now, that's going to make the DNA available or unavailable. We can also then, given that we have DNA, we can start binding proteins to that DNA, and we can start the transcription machinery. Okay, in the same way that in prokaryotes we had regulatory proteins binding to, in prokaryotes, to an operator, well, we also have regulatory proteins that bind to DNA in eukaryotes. We call these regulatory proteins, transcription factors, and the places that they bind to, control elements. Now, big fundamental difference. Here is a eukaryotic gene. You will notice there is no operator. Big fundamental difference between prokaryotes and eukaryotes is that all of the regulatory components in a eukaryote are upstream. They're either close in that they're called proximal control elements, or they can be large distance away. In fact, they can be on another chromosome, but they can interact with these other transcription factors to create our gene. The other thing to notice here is that there is only one gene. One promoter, one gene. In eukaryotes, that is, I believe, universal. Now, what do we have in the way of regulatory proteins? Well, we have general transcription factors and we have specific transcription factors. General transcription factors are part of the transcription machinery. Okay? Specific transcription factors, on the other hand, bind to specific chunks of DNA and act as labels and control mechanisms. So when we take a look at the general transcription factors, some of these are going to bind to the promoter, the Tata box, for instance. Some bind to our RNA polymerase, and together they create a transcription unit. Okay? General transcription factors are essential for transcription, but they are not specific for a specific gene. They're just the basic machinery. Now, all of those are going to essentially come together to form a initiation complex. Okay? That initiation complex is going to bind the RNA polymerase. It's going to produce your complementary strand of RNA, your transcript, and the regulatory component is going to come from those specific transcription factors. Now, specific transcription factors are going to be like the operator and its regulatory protein. Okay? But they're not going to block transcription. They're going to work together with these general transcription factors to produce that transcription complex, that initiation complex.
Most of the fine-tuning of gene expression occurs at the transcription level, making an RNA copy of a DNA sequence. Recall that to initiate transcription, an RNA polymerase must bind to the promoter region adjacent to a gene. This cannot occur without the help of specific protein transcription factors. Some transcription factors, called activators, bind to enhancer regions some distance from the gene. The DNA bends, bringing the activators closer to the gene, where the activators interact with other transcription factors and RNA polymerase to initiate transcription.